Okay, chapter 10, that's the characterization of nucleic acid and protein. So basically, in this chapter, we would discuss the manipulation of the nucleic acid and also the enzyme that involved that can be used to manipulate the DNA, and then for the further analysis. And also we would discuss the protein analysis. Okay. And then for to manipulate the nucleic acid, all right? And we have these two different types of the enzyme, okay? And we call it a nuclease. Nuclease, basically, they can degrade the DNA molecule by breaking the phosphodiester bond. And the nucleus can be the exonuclease or the endonuclease. For the exonuclease, basically, will degrade the DNA from the end of the DNA molecule and progressively move into the center of the DNA molecule. For the endonuclease, basically, will degrade the DNA from the internal phosphodiester bond. Okay, and for this endonuclease, it could be a sequence specific, which we will discuss further. Okay, and this is just to show you this. At the end of the DNA molecule, we have this phosphomonoester bond, all right? And this will be the target for this terminal the phosphatase, all right? The target for the phosphatase. And also we have internal the phosphodiester bond. That will be a nuclease target, all right? They will attack this internal phosphodiester bond. Right, see, remember this is 3' to 5' here, right? 5. This is 3, this is 5. Now, this is a phosphate. Connect to the previous oxygen and also this current oxygen here, this nucleus oxygen. And for the exonuclease, all right, there are two different typical exonuclease. First form we call it BAL31, right? BAL31 basically can degrade both 5' and 3', both double-stranded. So that's how that's why you see that the BAL31 can degrade both these 5' and 3', all right? 5' and 3' on both sides, on both ends of DNA molecule, and then progressively go inside. Eventually, will degrade all the DNA molecule, okay, completely. If the DNA is not bound by protein, then they will be degraded completely. And then another one is called exonuclease 3. Exonuclease 3, as you can see here, exonuclease 3 can only degrade at the 3', all right, the 3' and 3'. So therefore, you will see they will degrade from the 3', 3'. Okay, so that's the exonuclease 3. And then for the endonuclease, all right, endonuclease, all right, as I mentioned, it can be sequence specific. And for the sequence-specific endonuclease, then we call it a restriction endonuclease, right here, the restriction endonuclease. Basically, for this one, it's a sequence-specific base. It depends on the enzyme, all right? And then they will have a different recognition site, okay? And these two, the S1 nuclease and the DNS1, these two are non-specific. Okay, these two are non-specific. As you can see here, for the S1 nucleus, can only degrade the single-stranded region. All right, so if you have a single-stranded region here, or you have a single-stranded DNA region here, then it can be degraded. All right, but this S1 nucleus will not attack the double-stranded DNA region. Okay. On the other hand, the DNS1, okay, it can attack both single-stranded DNA region or double-stranded DNA region. So again, the DNS1 can degrade the DNA completely if the DNA is not bound by the protein, okay? So the DNS1. And then the restriction endonuclease, okay? So let's go to the next slide. Restriction endonuclease is sequence specific, okay? As I mentioned, it's a sequence specific nucleus, right? So they will basically degrade the DNA based on the recognition site. And the reason why we have discovered the restriction endonuclease is because the restriction endonuclease it derived from this bacteria's defense system. Because in the early days, in the 50s, scientists they observed, they found some bacteria. They are immune to the bacteriophage infection, and so from those bacteria, they discovered this host defense mechanism, because these bacteria they can they can produce a particular enzyme, particular endonuclease that can distinguish between the host DNA and the foreign DNA, which is the viral DNA, and then they will degrade the viral DNA, and that's how the scientists discovered the restriction endonuclease. So, for example, in here, from the cartoon that you see the bacteriophage infect the bacteria, then they inject the genetic material to the bacteria cell, all right? And then the restriction enzyme, basically, they can identify which one is the viral DNA and which one is the host DNA by these specific modification, because those host DNA are methylated, all right, are methylated, but the viral DNA are not methylated, so that the enzyme will recognize those DNA that are not methylated and then degrade that DNA, which we will discuss later. Let's go later on. We will discuss the restriction modification system. Okay, and then for the restriction endonuclease, all right. So there are three different types of the restriction endonuclease. We have type 1, type 2, and type 3. And for the type 1 restriction endonuclease, basically the cleavage site, okay, the cleavage site would be 1,000 base away from the recognition site. That's the type 1 restriction endonuclease. And then for the type 2 restriction endonuclease, which is the most important one, okay? And now we will discuss later. And type 3, right? Type 1, the recognition site and the cleavage site is 1,000 base apart. For type 3, basically the recognition site and the cleavage site is about a 24 base pair apart, okay? That's a type 3. So both type 1 and type 3, their recognition site and the cleavage site are not at the same place. Only the type 2 restriction endonuclease, okay? Basically, the recognition site and the cleavage site are the same for the type 2 restriction endonuclease. And the site, the recognition site, are typically inverse palindromic. So that means you can read the same forward and backward on the complementary strand. And many restriction endonuclease, the type 2, they recognize hexanucleotide target site, which means the 6th nucleotide. And here I give you some example. For example, the enzyme EcoR1, they recognize this 6th nucleotide, GAATTC, all right, GAATTC. That's a top strand. And then the bottom strand, if you read it backward, it's also GAATTC. So this is so-called inverse palindromic, okay? Inverse palindromic. So they can recognize these hexanucleotides. And then for ALU1, they recognize these 4 nucleotides. So AGCT and then backward AGCT. And then the PST1, you have CTG, CAG. And then you read it backward, CTG, CAG. Okay, so these are the recognition site for the type 2 restriction enzyme. And because they can be, the type 2 recognize and the cleavage at the same site, so it's particularly important, especially for the in the genetic engineering process, because when you process the recombinant DNA, you want to make sure you can cut the DNA at the precise location, and that's why we use the restriction enzyme 2. Okay, type 2 restriction enzyme, very important. And then from their digestion pattern, so you can see this red, the red arrow here. So the ECOR1 recognize GAA-TTC, GAA-TTC. The ECOR1 basically will cut the phosphodiester bond between G and A here. And here we'll cut it between G and A here. After the cleavage, then you separate into 2 molecules, right? From 1 DNA molecule to 2 DNA molecule. And because it cut between G and A, so they separate from here, all right? So 2 molecules. And then this AATTC, all right, that's a 5' end, right? And this AATTC button here is a 5' end. So we call this a sticky end, and there's a 5' sticky end, all right? This is 5' sticky end. For the ALU1, so basically recognize AGCT and then digest the phosphodiester bond between the G and C. So they cut it here, and then they separate into the 2 different molecules. And this is blunt end, right? It's called blunt end, all right, blunt end. And then PST1 recognize between A and G here, and they cut between A and G here. After they dissociate into 2 different molecules. So we have these 3', all right? So it's a 3' overhand, and this is 5' overhand, okay? 5' overhand and 3' overhand here, okay? These are example for a sticky end. It's either 5' overhand on the top here, or the 3' overhand, okay? And then the restriction enzyme also have different classification. So if they are isolated from different bacteria and they recognize this and cut the same DNA, all right? And these enzyme we call it isoschismal, right? Isoschismal, right? So for example, the BSPE1 and the ACC3, all right, they both recognize these sequences, TCC, GGA, TCC, GGA, and then they will cut here, all right? So these 2 enzymes, the BSPE1 and ACC3, will cause isoschismal. And some enzyme, they recognize, right? Recognize and bind to the same sequence of DNA, but they cleave at a different position. And we call these enzymes are neoschismal, all right? Neoschismal. With isoschismal, exactly the same. Neoschismal, that means identify the same sequence, but a different cutting pattern. So the example will be here, okay? So on the top, here's the enzyme NAR1. In the bottom is SF01. They both recognize GGC, GCC, right? GGC, GCC. They recognize the same sequence. But for the NAR1, they will cut between G and C here, and then separate these 2. All right. Okay? So this is the cutting pattern, and then they separate. So you will have 5' overhand, okay, for the NAR1. And then for the SF01, they cut in the middle here between C and G, Therefore, let's dissociate and 2 DNA molecule. This is blunt end, right? So this is a blunt end. Okay. And then another restriction endonuclease, we call it isocardamers. Here, isocardamers. Basically, they produce the same nucleotide extension, but they have different recognition site. Okay, like here, the example here. On the top here is NCO1. NCO1, they recognize CCA, TGG. All right, and then they cut between these 2C. So when you dissociate these two molecules, then you have this overhang CATGG. All right, it's CATG because cut here to here. So you will see the CATG. And then another enzyme is called PAG1. They recognize TCA, TGA, TCA, TGA. And also it cut between T and C. Okay, between T and C. After they cut T and C, then you get a CATG here, which will be the same as this. And we call these enzymes are isocardamers enzyme. Okay, so that's the restriction enzyme. And the restriction modification system, as I mentioned earlier, that for the bacteria cell to recognize which DNA is the host DNA and to recognize which one is the foreign DNA. So here you can see after replication normally, okay, after replication, then this A be methylated. All right, when they are methylated, then the enzyme will not be able to attack this methylated DNA. Even after the replication, initially you have this hybrid, one methylated, one unmethylated. We call it the hemi-methylated. Even hemi-methylated, like still can protect the DNA molecule from the enzyme attack. Okay. And so we call this a restriction modification system. And another type of DNA manipulated enzyme we call the polymerase. All right, polymerase basically are the enzyme that synthesize a new strand of DNA complementary to the existing DNA or RNA template. Okay, so basically like this, like a replication, you need a DNA polymerase. Okay, DNA polymerase, so you can synthesize this new strand. And the DNA polymerase, okay, DNA polymerase 1, basically DNA polymerase 1, they can find these. It was a single strand DNA region, all right, and then they can start synthesizing a new strand. And because it has the 5' to 3' exonuclease, so they can degrade the existing nucleotide and then put in the new nucleotide. Okay, that's DNA polymerase 1. So you can replace the original nucleotide and give it to a new nucleotide, but the DNA sequence is still the same. And clinal fragment, basically clinal fragment is like a part of the DNA polymerase 1, okay? And the clinal, basically, it's a part of the DNA polymerase 1, only has a synthesis function, all right? So that they don't have the 5' to 3' exonuclease activity. Therefore, with clinal-based replication, you can only fill in the missing nucleotide. You cannot replace the old nucleotide. Okay, it's clinal. And the reverse transcriptase, basically, you have the RNA template. Okay, RNA template. See the RNA template 3' to 5'. And then you can start the complementary DNA synthesis. And so that's called a reverse transcriptase, all right? In the presence of reverse transcriptase, then you can perform these DNA synthesis. you have to use the RNA as a template. All right, so you synthesize the complementary DNA. And then some other modifying enzymes. So for example, the modifying enzyme, the alkaline phosphatase, they can remove the 5' phosphatase, okay? That's alkaline phosphatase. Can remove these. Yeah, let me see. Who has a question? No, okay. The alkaline phosphatase, remember in the beginning I mentioned the phosphatase, right? They can attack these phosphomonoester bond, okay? So here, alkaline phosphatase, you remove this 5' phosphate group and replace with this hydroxy group, okay? And then polynucleotide kinase that will attach the 5' phosphate group. That will attach the 5' phosphate group so that, for example, after you remove this 5' phosphate from the alkaline phosphatase, then in the present polynucleotide kinase, then this hydroxy group be replaced by phosphate group. So you put it back here. Okay. And we have this terminal deoxynucleotide transferase that will attach deoxyribonucleotide to the 3' end of the polynucleotide in single strand or double strand manner. So it's here, you have this single stranded, so you add these to the 3' end. Right, so you can add this terminal deoxynucleotide transfer. You can add more nucleotide to the 3' end, okay? Either double, single-stranded or double-stranded. These are the DNA modifying enzyme, okay? And then another enzyme, the DNA ligase, right? DNA ligase, basically, they can repair this discontinuity because if the phosphodiester bond is broken, then you need to repair this, okay? Like this, ligase, or you can promote the two molecule to join together, okay? DNA ligase. Any questions so far? So that's the first part of the chapter 10. So we discussed those DNA manipulative enzyme, including the restriction enzyme, DNA polymerase, DNA ligase, and the DNA modifying enzyme.
There's no questions so far. Selena wrote one in the chat though.
Sorry, what did you say?
I said I have no questions, but Selena typed in a question in the chat.
Oh, okay. Let me see. Yeah, the background is a very broad. You can do the technology and also disease both. You have to explain, okay? Okay, now the application of the nucleic acid hybridization. Okay, so the hybridization, what does that mean? That means the formation of the hydrogen bond between the two complementary strands of a nucleic acid. And this is direct consequence of the stable double-stranded structure of nucleic acid under the physiological conditions. And this is due to the fact that the binding between the separate complementary nucleic acid is both reversible and the base sequence specific. Okay, that's a hybridization. So during the hybridization, okay, if both nucleic acid strands are not labeled with any isotope of fluorescence, but if one strand is labeled, then this labeled strand is referred to as a probe, and the process is called hybridization. And then one of these important application, right, the hybridization is the silent hybridization. So silent hybridization, silent blot, allow the detection of a given DNA sequence in a complex mixture of a DNA sequence. So it can be used to identify homologous sequence in genomic DNA or to facilitate gene mapping through the restriction mapping of gene. Okay, that's the silent blot. Okay, the first step, right, of silent hybridization is you purify the DNA, right? You purify the DNA. And then this DNA subject to the enzyme digestion, the restriction enzyme digestion. After the digestion, you perform the gel electrophoresis. After the gel electrophoresis, then you need to transfer the DNA to a membrane, and we call it the nitrocellulose membrane, all right? Because you want to have the use the membrane to perform the hybridization experiment. So you have to transfer. So after the gel electrophoresis, then you have to set up this DNA transfer from the agarose gel to the nitrocellulose membrane. And the setup is like this. They use the capillary the principal capillary principle to let the buffer go from the bottom to the top. During this process, they bring the DNA from agarose gel to the membrane on top of the agarose gel. So that's the transfer. So you transfer from agarose gel to the membrane. Then you fix the DNA on the membrane through the UV light or heat. And then after you transfer, then this membrane, basically the DNA is invisible. In order to visualize the DNA, you need to hybridize with labeled probe so that you can see the signal. Okay, so that's a silent hybridization. This is set up so you can see you have a paper towel on top of this nitrocellulose membrane and on top of this DNA gel. And then you have this paper that can draw this buffer upward. That's capillary, right? And during this process, DNA will be transferred from the gel to the membrane. And this is the result of the hybridization. This is another type of setup for the DNA transfer from the agarose gel to the membrane. So basically, you can set up these with the vacuum in the bottom, all right? And then you have the nitrocellulose membrane in the bottom. On top is the gel, and then you have buffer to cover the gel. So you apply the vacuum, vacuum. Then the DNA will be transferred from the gel to the membrane. Okay, there's different way you can use to transfer the DNA to the membrane. And the non-hybridization, basically, it's similar to the silent hybridization. And the only difference is the non-hybridization, you run the RNA instead of the DNA. Okay. And also during this process, you have to add the formaldehyde to the gel so that you can prevent the reformation of a secondary structure of the RNA. Okay.
Professor, question. So the northern hybridization, is it specific RNA markers? Is that what it is?
No, the sample should be RNA. You run RNA on the agarose gel. Okay, so the electrophoresis would be the same. Only here you run the RNA. For the southern blot, you run the DNA. Okay.
All right, so the southern blot, you run the RNA, the DNA, and the northern blot is for RNA.
You run RNA, correct.
Okay.
Okay. And then the DNA fingerprinting that developed from the southern blot. So basically, you cut the DNA under the study with the restriction enzyme, and then run the agarose gel and transfer to the membrane. Then you use a specific labeled DNA probe to identify the presence and the size of the fragments so that you can have this DNA fingerprinting. Okay. And another application of southern blot would be this restriction fragment length polymorphism analysis, RFFP. All right. An RFFP results from a variable number of tandem repeat in short DNA segment. Right. These VNTR, the variable number of tandem repeat sequence, can uniquely specify an individual homologous DNA sequence that can be detected by the presence fragment of different length after the digestion of a DNA sample question with specific restriction endonuclease. Okay, that's IFLP. So basically, the IFLP analysis is performed when the genomic DNA is collected and then digested with specific restriction enzyme, followed by the gel electrophoresis, right? And then the gel subject to the southern blot analysis. And then here, this example, let's say we have the parents, all right? The chromosome with wild type, the father with wild type, and the carrier. You have the wild type, the mutant, beta, the sickle cell, and mother also wild type and the sickle cell, all right? And the difference is the wild type is like this. You have the okay, this restriction site, and then the mutant here, all right, the mutant here. Then you have no restriction site because of the mutation, the single nucleotide mutation, right? And once this is mutated, then this is no longer available for the enzyme to recognize and cut, all right? And the wild type will still be cut, okay? And then for this, so that we know that the parents, both parents are the heterozygous. So one is the wild type, one is the mutant. And when you perform this IFLP, all right, this is wild type, this is mutant, then the parent will have one band for the wild type, one band for the mutant. And then for the children, so that if the children get both mutant, then you will see one band, all right? And then if it both are wild type, also one band here, all right? And then in between, you will have two bands here. Okay, that's the RFLP. And then of course the AFLP, okay, it's called amplified fragment length polymorphism. So you can simultaneously to analyze or evaluate hundreds to thousands of different DNA regions, distribute the randomly throughout the genome without prior sequence knowledge. So that's called AFLP. Okay, so let's watch the video briefly about the southern blot. Okay.
The southern blot is used to verify the presence or absence of a specific nucleotide sequence in the DNA from different sources and to identify the size of the restriction fragment that contains the sequence. In this procedure, the DNA is isolated from each source and then digested with a specific restriction enzyme. The DNA restriction fragments are then loaded onto an agarose gel and the fragments separated by electrophoresis according to size, with the smaller fragments migrating faster than larger fragments. The fragments are made visible with ethidium bromide. The DNA is then transferred from the fragile gel to a nylon filter. Next, the radioactively labeled nucleic acid probe is added.
So this is the probe. Okay, the probe basically is just the DNA that can hybridize with your target DNA sequence. And then the probe is labeled with the isotope or the chemiluminescence, which we'll discuss later. Okay, because the binding of this probe to the target, that will give you the signal. Okay.
The probe binds to complementary DNA segments. Note that the DNA segment being probed is not present in organism B. To detect the position of the radioactive probe, the nylon membrane is covered with an X-ray film. After development, the positions of the probe become visible.
So this is a typical southern blot procedure.
The southern
Then the detection method to detect the presence of the target DNA. Okay, for both southern blot and northern blot, the detection of signal is similar. Basically, you need to label the denatured single-stranded probe to hybridize to the membrane-bound DNA or RNA. And the probe must be labeled with a radioactive or other type of marker. And then denatured by heating and applied to the membrane in a solution of chemicals so that you can have a nucleic acid hybridization. Okay. And the probe can be DNA or RNA. And then the hybridization, okay, that give you the signal. All right, that's why we call it nucleic acid hybridization. We based on the complementary theory, the principle. So the probe can identify and find the complementary region and bind it, so that it give you the signal. And the binding of the probe to the target sequence depend on this melting temperature. Okay, the melting temperature, basically you can see this curve here. The melting temperature is defined as the 50% of the population is double-stranded DNA and the 50% of population is single-stranded DNA. So you can see DS equal to SS. At this point, the temperature will be a melting temperature Tm. Okay. And during the hybridization, normally you set the hybridization temperature would be like 1 degree or 2 degrees below the melting temperature, so that you can promote the binding of the probe to the target sequence. If the the hybridization temperature is too low, the hybridization temperature is too low, so we go this way. So that means you have more double-stranded DNA, and that would mean that you might have more non-specific binding. Okay, if you increase the temperature, then you have more single-stranded DNA, and that would also cause the problem, because the probe might reduce the ability to bind to the target. All right, so it's high stringency and low stringency. Okay, and so you have to adjust your hybridization temperature around this melting temperature to get the optimal conditions. Okay, and to determine the melting temperature, okay, so basically you follow this formula, right, for the solid blood, northern blot. So melting temperature equal to 81.5 degrees Celsius plus 16.6 log the molarity, okay, of the salt concentration for the hybridization, and plus 0.41 times percentage of GNC, and then minus 600 over the length of the probe. Okay, that's how you determine the melting temperature for the solid blood. And then, as I just explained earlier, okay, the stringency. If the temperature too low, right, then it's low stringency, and then it could give you the non-specific binding. If it's too high, then could have no result because there's high stringency. So you need to have the optimum temperature to give you the correct result. Okay. And then to label the probe, okay, the probe, you can label with the isotope. To label with the isotope, okay, there are three different ways. You can three different ways you can label the DNA probe. Okay, number one we call nick translation. Nick translation, basically, if normally DNA they have a nick, so you have this single strand DNA region. Okay, then you have the DNA polymerase 1. In the present DNA polymerase 1, then you have this p32D alpha ATP. So this is the structure. So you can see this ATP and alpha, all right? So that means alpha, beta, gamma. This alpha, this p32P alpha p32. So this phosphate is a p32 isotope, okay? So then you will just, in the present DNA polymerase 1, they will replace this existing sequence and with the new strand. So you will synthesize new strand. In the meantime, you incorporate this p32 DATP to the strand, and so the probe become labeled. Another one will be the clinal. Clinal basically is a fill in the gap, all right? So we call it the end filling, because you have EcoR1 sticky end, all right? And then you have the p32 DATP with the clinal. Then you can fill in this nucleotide in the end, and then it's labeled. Another one is random priming. Random priming, basically you have the hexamer, random priming. So that means you have a lot of hexamer, and then after denaturation, after you denature the double-stranded probe, then you hybridize with this random hexamer. And they will bind, and then they will have clinal fragment and the p32 DATP, and they can synthesize the strand and give you the isotope labeled probe. So these are three techniques normally people can use to label the DNA probe. Okay, and this is a typical result for solid blood hybridization. Okay. So after DNA electrophoresis, you transfer to the membrane, and then you hybridize with the probe. So you can see this. And on the right side here, that's the example of the Nolan hybridization. So you can see the signal. And normally this signal can be used to interpret the transcription level. Okay. And of course, you can also label the probe with the non-radioactive probe. Okay, so you can label with the biotintylated nucleotide, right? So you have this DNA probe, and then you have nick translation. You can use nick translation and filling or random priming. And then in the meantime, instead of using the p32 DATP, you use a biotin labeled DUTP. All right, in here, you use this alpha p32 DATP, this one. But here, you use a biotin attached DUTP. It's called deoxyuridine triphosphate, right? It's biotin labeled. And then from these three different techniques, then you you have this probe label with the biotin, and this probe bind to the target, all right, and then to detect the presence of the binding, so you have to add the avidin, right? Because avidin can recognize the biotin, and the avidin also is coupled with a fluorescence marker, and so that you can recognize the presence of the signal. That's one way. It's called the biotin-related nucleotide. And the other one is the horseradish peroxidase. So basically, you have a probe, and then you have single-strand DNA probe. Then you add the horseradish peroxidase and the glutathione. All right, so you will label these single-stranded probe, and then they will hybridize to a target. All right, and this horseradish peroxidase, then you add the lumina. All right, so they will have reaction and give you the signal. That's called chemiluminescence. Okay, so these are two different types of non-radioactive isotope probe and non-radioactive probe. Okay, and then the array-based hybridization. So this will be the beginning, the origin of the microarray, because from the stolen blood, so that stolen blood, we have a DNA on the membrane. Then you have the DNA probe. That the DNA probe is labeled either with the radioactive isotope or non-radioactive chemical, and then they can bind to the target and give you a signal. And from that individual hybridization, and then slowly they develop into this dot plot and slot plot hybridization. So for this dot plot, slot plot hybridization, basically, you can have a sample, all right? One sample applied to the whole membrane, and then use different probe, all right? Different probe to identify the presence of different probe. Or you can have each location, you have different probe, and then you have one sample, right, to hybridize this probe and to identify, either to identify whether the specific sample has the specific target, or in amount of 100 sample, and to identify which sample carry a specific target. All right, so that's a dot plot, slot plot hybridization. And from here, later on, the scientists develop this into the microarray. Okay, and dot plot, slot plot, just like this. For example, you have the DNA extract from different samples. There's 100 samples, right? And then you can plot on the nylon filter. And the way to do it is like this. This is a manifold here, all right? So manifold here, you have the membrane here, all right? And then you can each individual one, you can put one sample, right? So different sample, you can plot, you can put a plot here, all right? And then you denature by alkali, and then you add the probe, all right, the specific probe, because you have hundreds. Let's say here is 96 wells. You put 96 sample here. Then you put one probe to hybridize this membrane, and then from the signal, then you can determine which sample carry the specific probe. Okay, so this is one application for the top. And then the microarray. So microarrays, that's from solid plot, advance to the dot plot, slot plot, and then finally advance to this microarray analysis, right? This is the variation of the advanced technology from the dot plot, slot plot. So basically the dot material, right? Basically you have this material, which will be the probe. You have each individual probe. You put into the slide, right? You have the slide here, okay? And then you collect the sample. You have the let's say the reference sample and then experimental sample. You collect the sample, and then let's say you collect the RNA from the wild type, RNA from the mutant, and then you perform this reverse transcription, and you get a cDNA and label with different dye, all right? And that give you different color. You mix together, and then you put this sample to hybridize with the DNA probe on the slide, and these DNA probe, this DNA sample, basically they will compete with each other to bind to the probe on the slide. All right, and if a particular sample is more enriched, then they have more sample will bind to that particular probe. Okay, and that will give you a dominant color. And so after they bind, then of course you wash, and then you use the different laser to detect the signal, all right? And then that give you the signal like this. It's a red or green. Red or green, that'll be two extreme in both sides. So they say green. If you see green, so that means this sample here will be dominant. If the red, that'll be this sample will be dominant. If it's between, the color in between, then that'll be the depend on the amount, whether it's 30% to 40 to 70% or 45 to 55%. Then you need the computer to collect the data and then analyze it. Okay, so that's a microarray analysis. And let's watch the video about the microarray analysis.
A microarray is a small silica or glass slide, which is the size of a postage stamp, that contains thousands or even hundreds of thousands of spots. Each spot contains a synthetic piece of DNA with a known sequence. A microarray consisting of partial.
So these sequence, those are sequence here, that will be the probe, okay?
Sequences from a number of specific genes is created by placing a series of known single-stranded DNA sequences in an array on a microscope slide or a silicon chip. The sequences are placed in a specific pattern so that each position on the slide corresponds to a particular known DNA sequence. The portion of a microarray shown here only contains 6 spots, which are labeled A through F. These spots may correspond to DNA sequences found in 6 different genes. The expression of genes depends on the cell type and the environmental conditions. To study gene expression, messenger RNA or mRNA can be prepared from a particular type of cell, such as a muscle cell, or from cells that have been subjected to an environmental agent, such as a hormone. This mRNA that is expressed from such cells is used to make complementary DNA or cDNA. cDNA is made in a test tube by an enzyme called reverse transcriptase. This enzyme makes a DNA strand complementary to RNA molecules used as a template. Special nucleotides that have a fluorescent probe attached to them are used to make this cDNA. This probe will make the cDNA molecules fluoresce green when exposed to ultraviolet light. Once the fluorescent cDNA has been prepared, it is allowed to hybridize with the DNA samples spotted in the microarray. The cDNAs complementary to the type A mRNAs will hybridize with the DNA spot in the array that contains DNA complementary to the type A cDNA. Likewise, the type D cDNA will only bind to the DNA in the spot labeled D, while the type F cDNA will only bind to the spot labeled F. Since the cells did not express any mRNAs that correspond to the DNAs in the spots labeled B, C, and E, no cDNA is made that will bind to these spots. After hybridization, any unbound cDNAs are washed away, and the slide is examined under a fluorescence microscope, where the spots that contain fluorescent cDNAs can be identified. In an actual microarray analysis, a typical cell would express hundreds or thousands of different mRNAs, so that hundreds or thousands of different spots were identified. Therefore, microarray analysis is a powerful method to study the expression of many genes simultaneously from a particular type of cell or from cells that are exposed to a specific environmental condition.
Okay, that's the microarray. So till now, that's the DNA, RNA analysis, nucleic acid analysis. And then for the protein, okay, the most commonly used technique is called a Western blot. Okay, the Western blot, you can separate the protein from a complex mixture through this electrophoresis. And the protein separation basically is based on the size or the isoelectric point. Okay, but most of the time when you run the one-dimensional protein SDS page electrophoresis, is based on the size. If you run the two-dimensional protein gel, you'll be based on isoelectric point and the size. Okay. And then for the Western blot, basically you run the protein on this one-dimensional SDS page gel. And to run the protein on the gel, as you remember previously, the protein need to be processed. So to process the protein, basically, you want to make sure the protein will be evenly coated by the SDS, so that will carry the negative charge. And also, you want to denature the protein to make sure it would be it will show only the primary sequence, the single strand. No secondary structure or tertiary structure, only the primary structure. So you have to use the reducing agent like a DTT or beta mercaptoethanol. And then also apply the heat to denature the protein and to show the primary structure. And then you coat the primary structure with SDS so that the protein will be all negative charge. And then when you apply to the electric field, so they can migrate like a DNA from negative charge to a positive charge, and because they are all a primary structure, so and then coat evenly with the negative charge, so the migration distance will be inverse related to their size. All right, so that means the large protein will migrate slower with shorter distance, smaller protein migrate longer distance. Okay, that's the protein SDS page gel. And then after the electrophoresis, then you're going to transfer those protein from the chromide gel or agar gel to the membrane. Once they transfer, then the membrane, then you will process the membrane by, first of all, you have to block the, you have to do the block basically to reduce the background, right? That's why you treat the membrane with the BSA or milk solution. Okay, and then after you block the membrane, then you have a primary antibody incubation. So the idea will be similar to the probe to find their target, right? So the primary antibody depend on the specific protein that you want to identify, right? And then you let the antibody to find the target and bind it. And after that, okay, you have the secondary antibody, which is enzyme labeled antibody. Okay, it's an enzyme labeled protein A or protein G, and they will bind to the primary antibody. Okay, in this case we have this horseradish, right? Horseradish here, and then after the secondary antibody bind to the primary antibody, then you can perform this detection to see the presence of a specific protein. So this process is called the Western blot. Okay. And then the membrane, okay, for transfer protein to to the membrane, you can have diffusion through the capillary action, which is slower and not efficient. And you can also do the electroblotting or electrotransfer, use the electric to move the the protein from the gel to the membrane. Okay, and normally for the Western blot, we use the nylon membrane, which is good, and good the PVDF. These two are good. Okay, good strengths, but the PVDF give you a good staining. Okay, so these are the method for Western blot. Okay, and then another technique to detect the presence of specific protein, like antigen or antibody, that you can use the enzyme linker immunosorbent assay called ELISA. Okay, so basically ELISA is based on the concept of antigen antibody reaction. Okay, representing the chemical interaction between the antibody produced by the B cell of the leukocyte and the antigen. Okay, this specific immune response play an important role in protecting the body, that's the antibody, right, from the invade, such as a pathogen toxin. So basically, the ELISA permit a highly sensitive and selective quantitative and qualitative analysis of antigen, okay, including the protein or peptides. And there are two different types of ELISA. One is called indirect ELISA, the other one called direct ELISA, right? For the indirect ELISA, basically, you try to determine the presence of antibody. That's called indirect ELISA. So you have the recombinant antigen, or you have antigen, you purify the antigen. You coat it with the specific, the well that you perform the ELISA. And then you add the sample. If the sample carry the antibody, then that will bind to the antigen. And then you have the secondary antibody to bind this antibody. And later on, you can detect the color, the signal, right? So basically, they have this color developing stage that can give you the color, okay? So normally, for the ELISA, we use the ALP. Yes.
Professor, I have a question. So the
Yes.
direct ELISA is when you're taking the antigen and then exposing it directly to detect the antibodies, but the indirect ELISA.
Okay, no. Okay, let me. So indirect, direct. Okay. So basically, let's say you take the clinical specimen. All right. If you have the well that coated with the recombinant antigen or recombinant antibody, or even the antigen that you purify from the pathogen, right? So if you use the left side here, all right, the same clinical specimen, if you have antigen coated with the well here, then that will detect the presence of antibody, so that will be indirect. If it's on the right side, okay, if the well coated with the antibody, which can be a recombinant antibody, then you have a clinical specimen, you add it to the well, so only the antigen will bind to this antibody, and again that will give you the signal. So this is direct ELISA. And for these, let's say, for example, the easy example, let's say, for the COVID-19 virus, to determine the presence of the COVID-19 antigen in the body. So you, of course, that's the reason why it's so important to determine the sequence of the viral, the RNA, and from the viral RNA, and then you can engineer, genetic engineering to synthesize the S protein. Okay, they say the spike protein, and then you have a spike protein. Okay, then you can develop these recombinant antibody. All right, so you can coat it with the well, and then you collect the clinical sample, then you add it to the well, and then if the antigen is in the sample, they will bind to the antibody, and then later on you have this secondary antibody, right, that carry with the signal bind here, and that will give you a signal. Okay, that's the direct ELISA to determine the presence of the antigen. Indirect ELISA is to determine the presence of
Of
antibody.
the antibodies.
Marina.
Okay. No, I'm good. That I didn't hit the raised hand. I hit it before, but no, you clarified that. It was a little bit confusing, but thank you for clearing that up.
Ali.
I do have a question. So is the substrate already attached to the antibody that's already attached to, which then attaches to the?
You mean the substrate, the green one here?
Yeah.
The yellow attached to the green one, yes.
So that's already attached when you introduce it to the well.
This one, you mean the bottom one here?
Yeah, I'm
Yes,
sorry. Yeah, that's what I mean.
Okay, you have to attach this to the well first. So you have to pre-treat the well before you do the reaction. Okay. So like here on the right side, on the top panel, this is the direct ELISA. So you have this, you have to capture the antibody in the well first. Then you add the specimen. So if there's an antigen, they will bind here. Then you have this enzyme linker detection antibody to bind. And so that you can detect the presence of the antigen. So that's a direct ELISA. Indirect ELISA is to determine the presence of antibody. So you coat with this antigen here, right? And then in the presence, you add the clinical specimen. If there's antibody, they will bind. And then, of course, you have this secondary antibody with the enzyme so that you can see the signal, all right? So that's indirect ELISA. So remember, direct ELISA is to determine the presence of antigen. And indirect ELISA is to determine the presence of antibody.
All right, so the indirect is antibody, direct is antigen.
Correct. Correct. Very good. So let's watch the video, okay?
ELISA serologies are usually done in multi-well microtiter plates so that dilutions of serum can...
Yeah, because they use this microtiter plate. So that's why I say this well, each well, because you have this well here, all right?
ELISA serologies are usually done in multi-well microtiter plates so that dilutions of serum can be easily prepared and tested. To better understand how this assay is done, let's take a closer look at what happens in one of the wells of this assay plate. To perform the assay, the wells of the plate are coated with the antigen of interest. For commercial tests, this would be done by the manufacturer.
So if you have antigen, you coat here. That means you're going to detect what? What you try to detect now here for this video?
The sample antibody?
Antibody, yes, very good. So this is an indirect ELISA, okay?
Of the assay. To begin testing, the wells are filled with dilutions of the patient's serum. If antibodies against the antigen are present, they will bind to the antigen fixed to the bottom of the wells. But only antigen-specific antibodies will bind to the wells. The wells are then washed out to remove all unbound antibodies. Next, the solution of an animal antibody against human antibodies is added. This second antibody is covalently conjugated to an enzyme. The wells are washed again, this time to remove any unbound enzyme-conjugated antibody. Finally, a solution of a colorogenic enzyme substrate is added. The interaction of the substrate with the enzyme on the second antibody generates visible color. The development of color in the wells with a specific antibody can be seen with the naked eye or quantified with an electronic plate reader. The reaction is less intense as the serum is diluted and the amount of antibody captured in the wells decreases. The titer is the highest dilution with definite color development.
Okay, so this is the example.
So pretty much this titer tells you how much of an individual's antibodies are within a cell.
Right, correct. Okay, very good. So that's the end of chapter 10. Any question? Any question? No? Okay, so now we will continue this analysis of restriction digestive fragment because we just discussed the restriction enzyme. Okay, So the restriction enzyme mapping is very important technique. Basically, you digest DNA with enzyme, and then you resolve the fragment by the electrophoresis. Then through the number of bands that you observe on the gel, and also the size of band, then you can determine the number of the restriction site and the distance between the restriction site. Okay, this is normally done by, for example, if you purify the genome from the any, for example, pathogen or unknown pathogen, and then you don't know their sequence, then this would be the first step. You try to do the restriction enzyme mapping, and from the mapping result, then you can determine or develop or design the sequencing technique. Okay, and so let's go here. All right, so restriction mapping. Let's say, for example, here, this is a lambda DNA molecule. We have 49 kilobase long. All right, the lambda DNA, and then we have this bagel 2, BNH1, and SAL1. These 3 different enzymes. And then for SAL1, 2 sites. Okay, you have 1 and 2 sites. For the 2 sites here, because the linear DNA, all right. So if linear DNA, if you have 2 sites, then they produce 1 fragment here, 2 fragment, and 3rd fragment. So you have 1, 2, 3, right? You have 2 cut sites. For the linear fragment DNA molecule, you produce 3 different DNA fragments. And then for BNH1, the green one, BNH1 5 sites. For the 5 cutting sites, then you can generate 6 DNA fragments. All right. So here, see, you have 6 DNA fragments because you cut 1, 2, 3, 4, 5. After you cut it, then you produce 1, 2, 3, 4, 5, 6 fragments. Okay. So that's why it's a number of a band. Okay, can give you the how many cut sites. So let's say for this one, the circular DNA. This is the example of linear DNA. For the circular DNA, all right, the size of this circular DNA, if you run this DNA, linear size of DNA, give you 4 kilobase, all right? If you cut BNH1, 1 fragment, 1 cut site, 1 fragment, okay, 4 kilobase. If you cut with SHO1, 2 cut site, all right, SHO, then they produce 1 fragment here is 1.2 kb, and then another fragment here is 2.8 kb. SHO1, so you can see 2.8, 1.2. This 1 fragment, 2 fragment. Okay? And then if you perform the double digestion, so that means you cut the DNA with these 2 enzymes simultaneously. Then you will produce, because you have 1 cut site here and 2 cut site here, then you produce 3 fragments. So it'll be 1.1, 1.2, and 1.7. So you can see here, 1.1, 1.2, and 1.37, 3 fragments. Okay, so these look nice and easy. Okay, but later we will come to some exercise. Okay, we will do this later.
Restriction enzyme digestion is a fast and inexpensive way to gather broad sequence information about plasmid DNA. A restriction digest can give a generalized, bird's eye, or macroscopic view of the entire plasmid. It can also reveal useful information, like the purity and conformation of a plasmid, which is important for downstream applications, like transfection. For example, supercoiled DNA has higher transfection efficiency than nicked DNA. While in theory and in textbooks, restriction digestion is straightforward, interpreting the results of a digestion practice is easier said than done. In this video, we will digest a plasmid of known identity with restriction enzymes, run the products on an agarose gel, and analyze the results. We'll focus on identifying the bands produced under different conditions and use the results to make conclusions about the plasmid. Now, let's start our analysis. The first thing you should do is draw out your expected results. While this step may seem unnecessary, it's good practice and it will make your analysis go a lot faster. In this exercise, we would like to verify the identity of a plasmid we received from AdGene. We'll digest the 7.5 kilobase pair plasmid individually with either AGE1 or XBAL1, which are both single cutters, and then with AGE1 and XBAL1 together. We'll also digest the plasmid with EchoR1, which has 2 recognition sites in the plasmid. So let's draw out the expected results. Here's our gel. Lane 1 is the size marker, often referred to as the ladder. Lane 2 will be the undigested control. Because this plasmid is circular, the migration pattern of the undigested control diverges a bit from the migration pattern of the ladder, which was made with linear DNA. The uncut plasmid typically adopts a supercoiled conformation. That's because these plasmids were isolated from bacteria, and supercoiled is the native conformation of plasmids in vivo because it's compact. A compact conformation allows the plasmid to migrate through the gel at a faster rate than its linear equivalent. This is because it experiences the same amount of electromotive force, but less resistance due to friction. As a result, it runs a little smaller than the equivalently sized band on the ladder. This plasmid is 7.5 kilobases. So let's draw the supercoiled band right below the 7.5 kilobase pair mark. Another conformation of uncut plasmid DNA is nicked. DNA can be nicked naturally in vivo through the activity of topoisomerases. DNA can also get nicked during purification if the conditions are harsh. So it's common to see nicked DNA in your uncut control. When one strand of the DNA is nicked, supercoiling is released. So the conformation of nicked DNA resembles a floppy hula hoop or a hair tie, making it relatively large. In fact, nicked DNA is so big that as it's migrating through the gel, it experiences greater resistance friction than even the linear DNA, which can be more flexible than the nicked. Consequently, nicked DNA runs a little higher than the equivalently sized band on the ladder. Since we're still talking about a 7.5 kilobase pair plasmid, let's draw the nicked DNA a little above the 7.5 kb mark. Now that we've drawn our control, let's move on to our digestions. Lane 3 is the AGE1 digestion, which yields a single product. And because it's linear, its migration is consistent with a marker. So we expect to see a band at the 7.5 kb mark. Lane 4 is the XBAL1 digestion. Just like AGE1, it yields a single linear 7.5 kb band. So we expect to see a band in the same 7.5 kb spot. Lane 5 is the double AGE1 XBAL1 digest. According to our map, we would expect 2 products, a 7.2 kb band and a 300 base pair band. Since these 2 fragments appear in a 1-to-1 molar ratio, we know the 7.2 kb band will be 24 times brighter than the 300 base pair band, because it is 24 times as large. This means that if we don't have a bright enough 7.2 kb band, we might not even see the 300 base pair band. Lane 6 is the EchoR1 digest. EchoR1 cuts in 2 places, so we expect 2 products, a 5.4 kb band and a 2.1 kb band. Okay, this picture looks complete. Let's run our experiment and see our results. Run your gel long enough to get good separation of the bands. Don't rush it. Our experimental data with our expected results. We've marked the ladder with the sizes of the markers. Lane 2, the uncut negative control.
So this is basically the experiment that you can see for the restriction mapping, okay? And now let's go to this question, okay, the exercise. Let's say from here, you see they have to run the gel electrophoresis to identify the fragment on the aqueous gel, then you can interpret the result. Now let's say if you don't see this bottom half, you only see the top, all right? You only see this aqueous gel electrophoresis result. So we know one linear DNA fragment, if it cut with E-qualifier, it produce two fragments. So that means there's only one cut site E-qualified in that DNA fragment. And then if you cut double digestion E-qualified with BNH1, you see the 2.5, 1.8, 1.2.5. Then you have to try to make to match this sequence from here. So from 3.7 2.3 to here, then you can try to combine this sequence together to identify which one is which. So here it's easily that I can see 2.5 and 1.2 that make us 3.7, so that we know these two fragments come from this fragment. And 1.8, 0.5, 2.3, these two are from this fragment. Therefore, we know that there will be one cut side of the BNH1 within this 2.3 kb fragment, and then another cut side will be within the 3.7. Okay, and then you perform another single digestion, so you see 0.5, 1.2, 4.3, and so that you know that 1.2, 1.2, and 0.5, 0.5, so that you know these two fragments must be at both ends, and this 4.3 must be in the middle, right? So that's how you arrange this restriction cutting site. From here, we know equal R5, one cut side that give you two fragments, 3.7 and 2.3. And then from this single digestion to the double digestion, and then we found the 2.5, 1.2 must be from the 3.7. So we know BNH1 must be here, all right? And then I'll give you two fragments, 2.5, 1.2. And because we don't know which one will be on the left side, then you look at the single digestion, you see the 1.2, so that you confirm 1.2 must be here, and 2.5 is in the middle. And then meanwhile, you look at the other side. So 2.3 is a combination of 1.8 and 0.5, all right? Right, 1.8 and 0.5. So you know this side you will have 1.8 and 0.5, but you don't know which one will be on the edge here, right? 0.5 or 1.8. Then you look at the single digestion of BNH1, you see the 0.5. So that means 0.5 will be on the edge here on the right side. Then the 1.8 will be in the middle, all right? And so this is how you construct the mapping, the restriction mapping, okay? And then another example here, okay? So for example, this restriction mapping again, okay? Single linear DNA molecule, you cut with SBA1, give you two fragments, 24 and 24.5. And then if you cut with SHO1, give you two fragments, 15, 33.5. If you have double digestion, SBA1, SHO1, give you three fragments. It's 9, 15, 24.5. So based on these three data, let's try to resolve where are the enzyme location. Where are the enzyme location? First of all, look at the single digestion SBA1. Like give you two fragments so that you know there's one cut site for SBA1, right? So 24, 24.5. And now we look at double digestion, three fragments, 9, 15, 24.5. 24.5, 24.5. So we know this fragment is untouched, but the 24 fragment here has been cut to 9, 15, okay, in the presence of SHO1, so that we know this fragment, right, must have the SHO1 cut inside to give you 9, 15. And now we need to know which one, 9 or 15, would be on the edge. So then we go back to the single digestion here. We see 15 and 33.5. Therefore, we know 15, 15. So that we know 15 will be on the left here. So see, 15 will be on the left here, all right? Then SHO1, then SBA1. So this will be the arrangement for SHO1 and SBA1. And then, of course, you can interpret the KPN1 as well. Okay, so that's a restriction mapping determination. Any question?